Before we begin, I'd just like to note that this is the final RIDE seminar of the 2025-2026 season, and we hope that you'll join us next season, which is going to begin on September 9th with a presentation by Dr. Jaime Modiano, who is the recipient of this year's Zoetis Award for Veterinary Research Excellence. So please stay tuned for the full lineup for next season, and now it's my pleasure to invite Maxim Chirin to introduce our speaker.
Hey, welcome everybody. Well, and thank you all for staying back and to listen to the seminar. It is my pleasure to introduce Dr. Srinand Sreevatsan. Dr. Sreevatsan is the Dean of the College of Veterinary Medicine at the University of Missouri. And he's an internationally renowned and recognized scholar in infectious diseases, molecular epidemiology, and among other things. His research focuses on understanding how pathogens such as tuberculosis and influenza emerge, evolve, and spread across animal and human populations. He's authored nearly 200 publications, a lot of publications, secured federal funding, and is deeply committed to developing the next generation of scientists and advancing interdisciplinary research. So we're going to hear from him today about charting new paths, and I'm assuming you're going to talk about your path in doing research here across your career.
Yes, that's exactly what I'm going to do.
All right.
Thank you, Max. Thank you, Max. Can everybody hear me okay? I have a tendency to kind of swallow words sometimes, so if there is a problem understanding me, please shout out, and I'll repeat what I said, okay? I like to walk around. When my leg was better, it would have been easier. Now you all can have a good laugh at my limp, but it's okay. I'll take it in my stride, so to speak. So I'm going to briefly share what my journey in science is. And one of the big things that needs to be happening before I share the journey is some of the significant things in my life that has led me to be successful in these types of activities. The first person I'd like to thank she's somewhere in the room, my wife, Anjali. Congratulate her first, because of her, I'm standing here. And then I asked Gemini to tell me, because I couldn't list all the names of my collaborators from around the globe on this particular chart, so I asked Gemini to generate one through my Google Scholar page. And this is my collaborations across the globe in mycobacterial diseases, right there. There's mycobacterial diseases in there, and that's a big circle. That includes Mycobacterium tuberculosis complex, Mycobacterium avium complex, Mycobacterium paratuberculosis as well. So I worked on all of those, and I worked on chronic wasting disease. I'm not going to share the stories of all of these different things that I have done today. To be on time and be focused, I'll talk only about mycobacterial diseases, and an entire world to thank for that, including some of my mentors and everybody else who has helped me get to this stage in my career. So I was born in India. Oh, you could all probably tell. I went to school in the city of Bangalore, and then got my BVSc and MVSc, Masters in Veterinary Science. I specialized in canine dermatology in that particular period. Was a veterinary practitioner. Most of my cases were seen under a tree in a village, and I would treat mostly large animals. And then I moved to, well, there you go. I think I'm hitting the wrong button. That's the city. It's the southern part of India. And very humble beginnings, most fascinating formative years. Went to vet school. That's the front of the vet school building. It looks great. There's a little tree in the front here where I was a day scholar, so didn't have a hostel to go back to. I would sit in front of under that tree and do all my studying. So that was my journey during that time, and this is the capital of that city of Bangalore, and the garden, which we call as Lal Bagh, the red garden, lots of flowers, which are red, roses, etc. Beautiful place to go for a run in the morning. And you all will say, what the heck is this guy doing in this picture? I was the captain of the cricket team in my college. And guess who that is? It must be an old picture because it's black and white. That was me right there. And I did do a lot of sports, and you know, if this is an indication of something, it was not an injury due to some other stupid moves, but it was stupid moves during a game of badminton. Then I came to University of Minnesota. Minnesota opened its doors, provided me resources, and welcomed me with open arms. The first part of my journey in Minnesota was at the School of Public Health in the Division of Epidemiology, where I earned my MPH under the guidance of Dr. Ashley Robinson, who led the epidemiology program at the CVM here. And then I moved to College of Veterinary Medicine to pursue a PhD. Dr. Robinson, I tried really hard to find his photo. Couldn't find it. Anywhere on the web. Gemini couldn't find it. ChatGPT could not find it. So that's why there's no picture of him in here. But to tell you the truth, I mean, he just opened the doors and helped me get to start the program over here. At the time when I was finishing my MPH, he suggested that I should pursue a PhD. And I did. Didn't know where I was going anyway, so I did. And another hallmark event that happened was, some of you may remember Dr. Mather Kannan. He and I were talking one day, and he said that I should go talk to Dr. Sam Maheshwaran. And I said, yeah, I would, and I was continuing to work in the laboratory. Sam came, put his face into the lab door, and he said, Sri, come talk to me. Thank you, Sam, for doing that. That one event changed it. I changed over to Sam's group. Sam and Trevor were a team running bovine respiratory disease complex research. I worked with Sam developing vaccines for Mannheimia hemolytica as we know it today. During those days, we called it Pasteurella hemolytica. So then I got very strong beginnings in research, working under very strong mentors. And then I moved to Baylor College of Medicine, where I got exposure to true molecular mycobacteriology with Jim Musser, who was a up-and-coming clinical pathologist. He's a physician scientist, and he was able to direct me in areas of translational pathways. He was able to tell me what would take me and make me a lab rat versus what a lab rat could do to inform science and technology in medicine. So having done all of that stuff, moved on from there to work at a company called ClinSight. Some of you may know this person. He invented PCR, Kerry Mullis. He was a colleague in this particular adventure that I went through. Him and a few others who were running a company to develop diagnostic tests on a CD-ROM platform. It sounded great, but wasn't working well. So I moved on to go back to the academia. And once I did that, I was able to, well, where the heck is this going? There. So before I go into details about my journey in research, remember how I said Jim was able to tell me exactly how to position my own ideas and my own pathway in research. Jim showed me the Pasteur's Quadrant. All the epidemiologists in the room will understand this 2x2 table. There you go. There's a 2x2 table. We talk about the Niels Bohr quadrant, which is pure science, science for the sake of science and understanding of how things work. And Edison's quadrant basically talks about anything that's useless is out, only useful things will move forward. Pasteur's quadrant is performing fundamental science with use as being an important feature in whatever research we do. And that's where I spent most of my time. That is my quadrant, really. And you'll see why I say that when I go through some of the data with you. So you all know this. The higher education is now being targeted, right? So in exchange for trade schools, et cetera, we have to make sure, as those who are involved in higher education, educating future generation veterinarians, that we actually make sure everything we do falls in the Pasteur's quadrant, and those impacts are shared with the public so we can maintain the impactful activities that we have established in the academia. So this is where I talk about all of the different things that we do. Many different diseases have been worked on: prions, tuberculosis, Yarnies, MRSA. These were some of these programs began as collaborations with folks such as Scott Wells, Jeff Bender, and then with flu, Carol Cardona, and before her, Marguerite Papaioannou, who was at the School of Public Health. And prion diseases, generally, I brought that particular program from the industry where I was working on diagnostics. So I'll talk about the population genomic frameworks and biomarker-based diagnostics for Mycobacterium tuberculosis complex. This particular complex group of organisms include multiple variants like tuberculosis strictsans, bovis, capre, et cetera. All of those have 99% or more DNA identities. So which tells me that they should all truly be one species, but they are host specialists. And some of the things that we want to know is, how did they become host specialists? And can we use that information to help future in diagnostics? So we'll talk about the population genetic frameworks. That'll be the second part of my story. And the first part of the story is going to be about the biomarker discovery. So all the folks that worked on mycobacterial diseases, this is just a snapshot of a few that contributed to the stories that I'm going to tell you. Me Too Set began the first program in my laboratory working on proteomics from serum of infected animals to discover potential biomarkers for tuberculosis infection. And that was taken farther by Harish and Elise, who reinvestigated the ability to pull out mycobacterial proteins from that proteomics run. And Elise created some monoclonal antibodies to a few of those biomarkers, and we were able to validate that with an NVSL database of sample sets that they had. Sylvia Wanzala, she was doing an MPH in our public health program over here. She arrived one day in my office and asked for an opportunity to pursue a PhD. And she contributed to the understanding of not only the genomic frameworks of tuberculosis in the region of Uganda, she also contributed to understanding biomarker distribution for tuberculosis in deer populations. I'll talk about that. Deepti Joshi was one of the first ones who did a lot of genomic analyses with Mycobacterium tuberculosis. These are all isolates that are in the freezer. I'll tell you why we have to do it this way. Because it takes a long time to grow these bugs. And she established that and did some T-cell interactions with Mycobacterium tuberculosis complex. Much of her work was done at the National Animal Disease Center in Iowa. And Evan Brenner, accidental find, he came to my laboratory through the MyCab program here, and then went on to take a leave of absence. How many of you remember Philando Castile's event around here, right down the road from St. Paul campus? He was sitting at his window studying for oral prelims when that shooting happened. So you can imagine the consequences of that kind of an event when somebody witnesses it. And he finally got over it. And I'll present the story that he worked on in my laboratory at Michigan State. Joe is working on biomarkers. in the Michigan cattle and deer populations right now, he's validating the biomarkers, creating point of care or animal side testing. Alum, a Fulbright scholar from Pakistan, did a lot of work on biomarkers, revalidated the proteomics done by Me-Too, and then revalidated the work that was done by Elise and Sylvia, and expanded that work to the Michigan ecosystem. Michigan is living the outbreak right now of bovine tuberculosis. And Sarath Paudel, he came to my laboratory from Nepal. Actually, he was in Japan, Hokkaido University. He came to my laboratory, and he wanted to do some genomic analysis. I had him work on elephant tuberculosis, which is what his area of expertise is. I'll share a snapshot of his studies. And Preeti helped a lot in Mycobacterium paratuberculosis work, and so did Fernanda, Sajani, Alifia, and Abhirami. They were all very productive. They all published at least 2 papers before they graduated. Remember the keyword, at least. But that was not my requirement of them. So tuberculosis is important. Why study tuberculosis? It's a disease of poverty. It's a disease of people coming together in crowds and hanging out together. But remember, even then, it is about 1/3 of the world population is infected with tuberculosis, exposed, infected, and some actually progressing towards disease. You need to know this. And why study tuberculosis? If it's a human disease, how does it matter for veterinarians? Because these are highly specialized organisms with tremendous capacity to transmit across species. Some people call it zoonotic infections, et cetera. I like to call this interspecies transmission, because it can go seamlessly from humans to animals, animals to humans, regardless of the variant. But there is a period of optimization that these organisms go through. And I'll tell you why when we talk about Evan Brenner's work. And then, of course, drug resistance is a big deal in tuberculosis. That was one of my first projects in Jim Musser's laboratory. At that time, 1995, yeah, it's the previous century, 1995. There was no knowledge at that time about any kind of mechanisms of drug action or targets of drugs in Mycobacterium tuberculosis complex. The first paper on the target for isoniazid was published in Science by Bill Jacobs and his group. And the rest is history. We went on in collaboration with Dr. Bill Jacobs, we worked with Dr. Cliff Barry, identified multiple targets for all of the frontline TB drugs. And we studied those. And I'll present some of the ideas around why we need to understand that to be able to make good decisions that'll help our patients. So first is, sometimes I said, we have to work with freezer stocks, right? That's a bias, of course, but we have to understand the population genetic frameworks of any pathogen before we start asking questions about, is there a blip in a specific genotype in a specific population? So population genetic frameworks based on about 6,000 genomes of tuberculosis complex basically classifies them into 9 lineages, the first 7 being human TB strict sense. Depending upon their geographic origin, they're classified as Indo-Oceanic, sorry. I gotta go back, how do I do that? There are lineages that are identified based on the geographic origins of these different lineages. West Africa lineage, the Indo-Oceanic lineage, and then there's the Euro-American lineage, et cetera. I'll tell you why that is also important when we talk about the elephant tuberculosis. There, so some of the first stories, circulating biomarkers in tuberculosis, can they be informative to us in identifying disease, infection, not exposure to infection. The TB diagnosis today is basically an exposure to infection. Can anybody tell me what's done? Have you Some of you have worked in BSL-3. Well, nowadays we do gamma gold or gamma interferon release assay, which is basically telling the T cells to recognize specific TB-specific antigens and respond to it by releasing gamma interferon. And if we do that, it's only measuring a response to exposure. Same principle, which is applied in animals, caudal fold test. There was a test similar to IGRA or the interferon gamma release assay. It was called Bovigam that was developed. It has been sunset, but the kits are still available if you all want to try it out at some point. And so this was followed by by a second round of studies by Me-Too Seth that said that Me-Too basically concentrated on defining host biomarkers, host molecules that are responding to infection in the animal. And Elise basically concentrated on pathogen-specific molecules that are released into circulation during an infection. So remember this, if there is infection, you will have mycobacterial origin peptides in circulation. If there is no infection, no peptides. Is it better than measuring gamma interferon release? Think about that for a moment. So all this work was done in collaboration with my partners, Ray Waters and Mitch Palmer at National Animal Disease Center. They had an experimental animal model. They had followed over a period of 6 months. They had a baseline collection of blood followed by exposure to bovis and then subsequent monthly collection of serum. That gave us a good longitudinal design to identify age-controlled biomarker release in these animals as infection progressed. It was actually highly informative for us to understand this. This was then taken on by Sylvia, who published a paper in Deer Tuberculosis, which is down here. So Me-Too, when she came to my lab, she first volunteered for 2 months in my laboratory to just learn her way around the lab and learn the techniques and everything else. She applied to the CMB program. She was denied admission at the CMB program. So she tried the Vet Med program, and they said, okay, we'll let you in for an MS. But she is a topper from her vet school, and she had done some really good stuff. So when she came and asked me to take her on in my laboratory, I said, yeah. And she did. She went by herself to all these seminars on campus. One of them was in the cancer center where she heard about biomarkers using eye-tract analysis and peptidome hypothesis. This is what oncologists are looking at, pancreatic cancer detection, et cetera. It's all based on this particular hypothesis. Using this hypothesis, she performed the proteomics with eye-tract, the multiplex system, using the NADC collection that I talked about. So the initial identification happened, and then followed by ELISA development. We had to create monoclonal antibodies against those peptides we had identified. Develop an ELISA. Yeah, if we put a peptide against a monoclonal antibody, you'll get a perfect result. But if I challenge it with field serum samples, will I be able to get the same kind of a readout? That kind of validation was done much later, after we had fully identified all of the peptides that we could find, and then identified cross-reactivity with other mycobacterial exposures, Johne's disease, Mycobacterium kansasi infection, and several other co-infections that would be potentially cross-reactive with the peptides we were pulling out from the proteomic analysis. And then the other question was, what about white-tailed deer? They are probably reservoirs. Once it escapes from the domesticated animal species into wildlife, TB has a tendency to maintain itself and reappear every now and then. So this actually became a very important question. I'll take you all back to 2007, about 20 years ago, when there was a bovine tuberculosis outbreak in the state of Minnesota, in the northwestern part of Minnesota. And there was a need for more robust testing and be able to give an opportunity to look at ideas other than depopulation. Funding was limited. It was bovine tuberculosis, after all. NIH wouldn't even look at us. So we connected with Michigan State. Scott Wells and I collaborated with Michigan State that had a line item dollar coming to them from the USDA to respond to tuberculosis. Scott and I wrote the grant. Scott wrote the epidemiology piece. I wrote the biomarker piece. It was funded. Actually, it helped me do one round of proteomics, about $30,000. But it was enough to revalidate. It was not greedy to get more. Once we revalidated, Sylvia was able to take it on and move it to the next phase. And of course, the Department of Natural Resources came into play here. I tried hard, finally found a line to reach Dr. Michelle Carstensen, who was at DNR at the time. I think she's still there, right? Yeah. So she was able to share her full repository of 384 plus naturally infected deer samples or exposed deer samples from their sharpshooter collection. The helicopters that went out sharp shot, they collected blood, they had large amounts of it. They gave us about a 1 ml aliquot. I still have several microliters of that left. So after all the validation. So new biomarkers, I can use the same sample sets to revalidate. And then I asked the question, does it matter if I test these biomarkers only, will it work only in the bovine host and the deer host, the cervid host, or can I apply it to a broader range of MTB complex infections across the globe that would include tuberculosis, strict sense infection in humans, in primates, and in other animal populations. Since they share so much in nucleotide and proteomic identities, it is possible. But pathogenesis is different. Host specialization has led to many internal optimization of polymorphisms in moving cholesterol from the host into the bacteria. And the availability of cholesterol is different in different host species. So how do you do that, right? So it was actually an interesting undertaking. We knew that there were challenges. We still tried. Okay. This was a part of a graph. One of Scott Wells' students, Joao Lima, helped me do the analysis. He simulated my data from the 500, 600 cattle samples that I had tested, and he binned them into infected, true infection, where we had culture information from those animals, exposed, if they came from the same herd, but uninfected, because they tested negative on the caudal fold test. And then he had pristine samples which were unexposed, uninfected from other parts of the country where we had collected, where there is no bovine TB. But depending on the prevalence of the disease, he simulates the sensitivity and specificity in this particular graph. So depending upon what the prevalence is of the disease, if it's very low, where do you put the cutoff point? About probably 0.2, 0.3. If it's very high, probably 0.4 in that range. There are some comorbidities that may cross-react. So experimental deer data also showed extremely good biomarker. Let me come back here. I know, wait. Oh, I'm going forward. Now I'm up. Come back. Yeah, there's that one biomarker that's PKS5. There's the other biomarker, MB1895C, basically simulated over prospective collection of information in experimentally infected deer sera. And this was the work of Sylvia Vanzala. And then she did the work with MB2515C as well. These three biomarkers were then challenged with the same ELISA, same monoclonal antibody on the field sera we had from DNR. Clearly, they were all testing positive. These were the animals which had come here to the diagnostic laboratory for necropsy. They had a granuloma. The granuloma cultured positive for M. bovis. And those were the only ones that were used in this particular analysis. We asked the question, does it work in primates? if they have TB. So I approached my collaborator in Tulane University in the primate center over there, where they were doing SIV infection, followed by tuberculosis infection to study the co-infection parameters that may contribute to pathogenesis and potentially inform treatments in the human situation. I was not interested in the HIV part, I was interested in the TB part. I asked him to send me some serum samples. He did. Dr. Tiffany Wolf got me some samples from Yerkes National Primate Laboratory from Emory, which were all negative for TB. So that's the baseline right here. These dotted lines there, those are the negatives that I got from 2 different primate institutes that had those samples. And the experimental infection basically showed similar trends in increasing amounts of PKS5 or 2515C or 1895 in the primate model. So what about humans? So I believe I collaborated with Dr. David Boulware over in the medical school. He's an infectious disease physician doing a lot of work in Uganda and other parts of Africa on Cryptococcus neoformans and many other HIV comorbidities. He was able to help me identify a way to do this with some funding. So we wrote a grant to the CTSA at the University of Minnesota. We were funded $50,000. I spent $150,000 getting samples from Find. And $50,000 were enough to buy the ELISA plates and run the ELISAs. And there you are. These are TB negative samples from Find versus TB positive samples from Find, and how they were testing consistently positive for all the 3 biomarkers we tested. One thing I learned from doing this biomarker testing and prospective analyses was probably need to use more than 3 biomarkers in the testing because they may appear and disappear as the infection cycle progresses. Sometimes they can be highly informative in response to treatment, especially in human tuberculosis. If the patient is responding, the bacterial load is reducing, you'll see a reduction in biomarkers that are specific to tuberculosis complex. Okay, so we'll change gears, we'll go to the genomic epidemiology piece. This particular paper, I mean, this is very interesting because it started as a collaboration with an institute in Brazil at the University of Viçosa, where I went there to do a small course in molecular epidemiology. On the way back, my host suggested that I stop in Fiocruz in Rio to be able to have a conversation with the Fiocruz people who worked on tuberculosis. I did. I met with this person here, Dr. Paulo Basta. Paulo Basta works on human tuberculosis, specifically in indigenous populations of Brazil. And he approached me after my presentation and said, hey, Sri, we would like to collaborate. I have access to TB isolates from a tribal population in Brazil where there was no treatment in the past at all. And I'm now trying to get them into the first-line TB drugs and be able to clear TB in that population, but it's more complex. So we do, when we look at the TB activity in Brazil, it's quite rampant, actually. It has about 3x higher incidence in tuberculosis compared to the rest of the area, and it's been increasing over time. And then you take a look at, in the state of Mato Grosso do Sul, did I say it right, Dr. Gaidas? Mato Grosso do Sul is the place where the tribal population lives, and there it is 6x higher. So it's a very high incidence region. Why is that? Or high prevalence region? And it's also because Mato Grosso do Sul is located on the Paraguay border, very remote location. And how did the tribals get this particular disease if there wasn't any disease in the past? Unless there has to be either a reactivation of tuberculosis or there has to be a new introduction. And we can test for both with genomes. And I'll tell you how we did that. but 60% higher in Mato Grosso do Sul. And the Guarani and Kiowa, the two tribes that populate that area, both the tribes had that. They were using only herbal medicines to treat TB. So it was interesting. We went and he sent me the full series, I think about 80 or so isolates that he had. We did the genome sequencing. And that was the work that was done by Anam Hadi, the Fulbright scholar who was visiting my laboratory. And then, because TB is a blind spot, when we did the lineage identification, interestingly, all the isolates that we had from Mato Grosso do Sul clustered in lineage 4. He said, hey, there's a point source outbreak going on over here. Wait. We go back and we do barcoding by 414 genome-wide SNPs that was established by Cole. It was a publication in Nature. We checked it out. We consulted with that author. It turns out that was actually a very robust approach. We took it, and then we took the SNPs, assigned the lineage, looking at the barcode activity. We had lineages 4.3, 4.4, 4.1.1.3, and there were at least 1, 2, 3, 4 different clusters of lineages that we identified. What does that tell you? It tells you either there were multiple exposures or multiple introductions into this population. But is that true? Can we test that by looking at drug resistance associated mutations within these genomes? And Anam did that next. Oh, sorry. See, that is the reference, and then there's the different groups that they clustered into. So when we look at drug resistance, I said, sure enough, there's drug resistance in these bugs. Pretty much every bacterium we were testing had a mutation in one or more of the drug resistance associated genes, suggesting a strong association to drug resistance. How does this happen in an isolated population? Clearly, this is an acquisition from an urban location where these people are going with their handicrafts to sell and to bring back money and other types of goods to their tribal populations. What I did not have in this particular study was the social network analysis within that population. Dr. Basta's postdoc, Dr. Ida Victoria Colty, had that information, but she hadn't done the full analysis. And Anam was on a timeline to finish her PhD, so she published and moved on. But the idea is still to overlay the social interaction networks on my genomic lineage networks and to be able to identify where the attributions were in terms of transmission that was going on in that part of the world. Very interesting. After that, I got a lot of emails from Argentina and other places to request how to go about doing similar type of activity. And I think everything was published and supplemental data had all the methods, so it was pretty easy to go after that. Well, these are all 42.1% is generally what we finally got. This is the conclusion slide that Anam put together. It was her PhD defense slide. She said it was the first TB sequencing study in Guarani Kiowa population. True. It was an isolated population. And then, of course, one dominant clade. There were at least 3 other dominant clades that were identified in the extensive drug resistance associated mutation study, which she did a fantastic job of. It was not easy to pull those mutations out from those 80 plus genomes. And then came gems in the genomes. We had genomes from Brazil, we had genomes from the United States, we had genomes from around the world. We had over 12,000 Mycobacterium tuberculosis complex genomes. The automatic idea is, oh, can we create a pangenome? I said, let's not lose the information by creating a pangenome. Let's ask the question as to how much variations there are within these pangenomes that can explain to us the host adaptation principles. So Evan Brunner undertook global GWAS analysis, and I tracked some people here who are geneticists. Dr. Mickelson, you will like what he did. This was actually a very well done study by Evan. He had, of course, there are biases here because not everybody isolates Mycobacterium bovis or animal granulomas don't go through isolation all around the globe. Only in certain pockets you get. So there's a geographic bias in the collection of isolates. And then the ones that do have those genomes don't want to share them. They don't put them in the NCBI, so we are basically lost at that point. So, but we were still able to get complete genomes from 6,000 different isolates and from pretty much every geographic location on the globe, and GWAS analysis was undertaken. One of the questions I had for Evan was, last century, 1996, I had discovered one polymorphism that separated Mycobacterium bovis from all MTB complex group of organisms. I asked him, is that still valid? He said, Sri, I'll give you more. And that's what he did. He did this GWAS analysis, and he asked about all of these important questions in the GWAS. And I won't show you a lot of Manhattan plots that he put together. It was GWAS done with what is called as bugwash. These are bacteria. They're not eukaryotes. They have a single copy of everything. And so you have to use different assumptions to do the GWAS analysis. And he performed it, identified the SNPs, and identified 120 concordantly identified mutations that clearly separated Mycobacterium tuberculosis variant bovis from Mycobacterium tuberculosis complex group of organisms. And that included that one mutation that I discovered about 30 years ago. Yeah. So indicative of host adaptation, it is possible because 120 loci that he identified. Remember I said cholesterol hijacking from the host? All of the genes are associated with cholesterol hijacking and cholesterol metabolism because they need that much cholesterol to be able to restructure their cell walls. Mycolic acids take a lot of effort to put together, and so they steal molecules from the host. And these pathways are all in that. So think about this. If you take M. bovis and put it into a mouse model, sometimes works, sometimes it doesn't. You take that and put it into a rabbit model, it works well. Put it back in a calf model, it's perfect because they know how to use metabolic pathways of their primary host. And so it was very indicative. Why are we interested in these 120 loci to separate M. bovis from MTB? There is a very basic difference in the way we can grow the two bugs. One requires glycerol, and the other, the glycerol is not a good carbon source. It's actually toxic. That is Mycobacterium bovis. So all these years we've been growing the bacteria on glycerol-containing media. How much information have we lost? Do we know how much, the extent of interspecies transmission that's going on, which species is infecting which host? Some of these things would be useful. Also, Mycobacterium bovis, or Mycobacterium tuberculosis variant bovis, is naturally resistant to pyrazinamide, one of the frontline drugs. And it is marginally resistant to isoniazid, also a frontline drug. So would that information be useful to the physician to make a decision on which way to treat this patient when they come to? We don't treat in the animal world. We just depopulate. But these are all useful pieces of information. They can also inform us about pathways of transmission among animals and humans. Then enter the last story, the story of elephants. So I wore an elephant tie, so I could share this one. The elephant is a flagship species on Earth. It's sort of the remnant of the mammoth. And it's one of those species that we want to protect and keep. And it's sort of its natural habitat is sort of restricted in Southeast Asian area, around Nepal, India, Thailand, Myanmar, etc. Indonesia, that's where you'd find all of these, most of the elephants that you see in the zoos have been brought from one of those places, and they're probably carrying tuberculosis. Well, that was a major discussion a long time back by the USDA as well. Dr. Sharad Paudel, a veterinarian from Nepal, who was treating elephants in Nepal, as well as collecting nasal washes for TB testing, came to my lab as a postdoc. what luck, he brought with him the isolates that he had collected from his necropsies. And he wanted to ask all these beautiful questions about, is there a non-random distribution? Epidemiologists tend to ask these types of questions. And are elephant TB isolates from Nepal genomically different from others from other countries? So he had to do an overlay of distribution of population, genetics of TB, with his own genomic analysis of elephant isolates. So we went to, his collections were all in Chitwan National Park or in Koshi Tapu Wildlife Reservoir. And one animal was identified with TB in that region and different parts of the Chitwan National Park. So yeah, very interesting area, lots of tourism there. People are coming in from all over the world, from China, from Bhutan, from India, from other parts of the world to this region for many, many reasons. One is to be able to do tourism. One is to see if they can actually do the Mount Everest, and then have an opportunity to meet the elephants. These are the necropsies. Sorry, it's immediately after lunch. I had to share a pathology slide here. The granula was a caseation. It's so typical. And the lung sizes are huge. So you can imagine the bacterial load that these animals will carry when they're going through tuberculosis. So he was able to easily get isolations out of these. And we overlaid our genomic analysis with the population genetic framework that exists of lineages around that particular region of Nepal. And you can see that the predominant lineage is lineage 2, the Beijing strain, the biggest example of a BCG vaccine, SKP, that actually is rampant in that region. And there are other genotypes that are lineages that are identified as well. Lineage 1 is usually rare. I'll tell you why that is important to remember. In that region of the world, and that is the Indo-Oceanic lineage. And when we look at the SNP barcoding, the same methodology as Cole et al. That was performed in the previous study was applied here. They were all lineage 1, they were clonal, except one in the Chitwan National Park that was lineage 3. That tells me that there's at least 2 introductions that have happened in that region into the elephant population. Also tells me that there is one particular isolate here that had 2 different lineages, so there's multiple infections with an animal that's possible, and we are able to detect it with genomic analysis. And they were all lineage 1, but see where that yellow little piece of the pie is, it's very small. So there is an indication that we could track back if we had the information of tourism in that area. These were all historical isolates, so that's a historical isolate bias. But it gives us an opportunity, establishes the baseline to go back in and do a longitudinal analysis, identify infections, look at the mahouts or elephant handlers, and do their testing as we go through this entire process. It takes a lot of funding. USAID doesn't exist anymore, unfortunately. So there goes that idea. Well, life beyond academia, I said thanks to my wife. She grounds me. Because of her, she was able to permit me to spend hours together in the university. And she basically is my rock that I revert back to. And that's my daughter. We like to, whenever we get an opportunity, run, do other things together. And of course, our life has gone to dogs. We own multiple dogs. These are the recent ones. I've had dogs for the last 30 years, and mostly all boxer breed. And they're all rescued. And of course, sometimes we have to spend our time at the back end of a cow. And this was the team in Michigan State University before I left there. Of course, it is not the entire team. This guy, this dude standing right here behind Sajani, his name's Mark Haman. He's a veterinarian from Nigeria. He's currently working on developing a oral delivery tuberculosis vaccine that could be applied to deer populations and potentially in cattle population. And that's his research. That's a different story for a different time. Major Glowacki, she's a DVM MPH PhD from Ohio State, came and did her PhD, sorry, DVM MPH from Ohio State, did a PhD with me in food safety. She's deployed back into that region where there's a lot of strain going on right now. And Kelsey Bauer recently graduated from DVM at Michigan State. So that's my talk. I'll take questions. I'll be happy to interact with others after this too.
All right, here's a hand.
So John Green is, a novelist who wrote the book about how everything is tuberculosis, and it is no longer a scientific problem, it is a political
Border problem, yeah.
Yeah. What is your response if you were being interviewed by National Public Radio? What, as a counterpoint to that, how, and as a true One Health scientist, what's your response to that?
In a sense, I don't want to say it in percentages. In a sense, I agree with that thought process. It is a political problem. There's restriction of movement and other things that happen from some of the developing countries. Even me, when I made my first move into the United States, I had to have my chest X-ray. I had to have my tuberculin testing. IGRA was not really popular in the last century. Yeah, it dates me, doesn't it? So I can see why he's taking that stand. And as a One Health scientist, I think we have to go a little bit more into the details of why that exists. We can't just go and tell politicians that they should do different things differently. We have to tell them why they have to do things differently to be able to take a global approach to controlling the disease. Well, COVID came and went. COVID overtook its global incidence and prevalence in 2020 from tuberculosis. In 21, after mRNA vaccines, that went away. TB still stays high, killing people. It's a wicked disease. It's a disease that it's an organism that has co-evolved with its host for millennia. So there's definitely a lot of work that needs to go into eliminating tuberculosis. There is a program called Eliminate TB. Politics are interfering, and I think that book was timely.
Any other questions over there? Hang on, Scott.
Scott?
Hang on, just a second. Yeah.
Zoom and recordings have to catch the question too. They could have repeated it.
Sri, your presentation has demonstrated a lot of collaborations, right? Team science, right? We've been trying to do this a long time. So what are some lessons learned? You're in a position now where you can put this in place. We're trying to do the same thing. How do we do team science? How do we do it successfully to get the Pasteur's Quadrant?
Yeah, team science is the key phrase I use as well in my current role about having people think about team science. I do think, in the clinical world where there are clinicians who have access to the most important and most valuable type of samples as patient populations, et cetera, collaborating with fundamental scientists to go into the Pasteur's Quadrant would be fantastic. Team science it sort of comes together organically. Sometimes we can put money in front of team science and say, like the one that we wrote to Michigan State to get money for, gave us $30,000. It was nothing, but it gave us enough to get started. That gave me the preliminary data to get the USDA, the big USDA grant, which then funneled into, rolled into a bigger grant and bigger collaboration. I just wish that there were more investments into these types of diseases. It's an interesting question to be thinking about team science. And we have talked about team science when I was here as well, only 10 years ago. You recall some of the research committee meetings where team science was actually a part of the discussion. Talk is cheap. We have to show it. It takes every individual to make that mind change to say, hey, look, I know collaboration means I have to do some compromising, that's fine. We don't, see, the system had set it up such a way that we recognize you for your successes. We don't care if you are a partner with somebody else on a grant. That's wrong. I think we should give recognition to every possible collaboration that comes together. Collaboration is this. Collaboration is not that or that. So team science has to come through that kind of a mindset.
Fantastic. One more question, perhaps, before we go to the awards. Max, hang on.
Me walking around is not very safe, I guess. I almost tripped him.
Yeah.
Sorry.
So Shri, I'll ask you a science question. Your work with elephants, it looks like it was an isolate that appeared to infect the entire region, except for probably one that was off, that was different, right? So there was
Oh, there's the elephant TB.
Elephant TB.
Yeah.
Any conjecture in terms of where that came from? Is that local? Is that there? Could it come from the human side? Or is this
All of them are Mycobacterium tuberculosis variant tuberculosis. None of them were M. bovis. All of them were tuberculosis strict sense. Definitely human to animal transmission here. Definitely. And maintained by the animal population. Speculation is the gift of a scientist. I can speculate the heck out of this, but I'm going to stay away from it.
Fantastic. Thank you so much.
Thank you.
We're going to proceed with ceremony.